Great. Thank you, Pretty. And thank you for having me. Let me get my laser pointer. I'm super excited to share our latest story on how neuronal signaling affect germline development during stress in C. elegans. And I'm leaving my blue sky handle here, so please reach out if you have any more questions after today. Cool. So no organism on Earth lives in isolation. The tight relationship between the organism and their environment is a universal feature of life. In many fishes and turtle, sex is determined by temperature, and global climate change is severely threatening their reproductions and survival. In social animal like honeybee, for example, their role in hive is determined by diet. The fertile queen is fed with royal jelly and grew substantially larger than the infertile worker. So it's therefore clear that the environmental factors play a significant role in organismal development. So it's important for us to ask, how do animals cope with the unstable climate that we're currently experiencing? And what is the molecular basis of gene-environment interaction underlying complex trait and diseases? The star of my presentation today is non-coding small RNA. When coupled to a protein called AGO, they can find genes that could pair with and cause post-transcriptional silencing, resulting in rapid reprogramming of gene expression. And this machinery may serve as an interface that integrate different environmental information and in turn regulate gene expression, allowing organism to launch an appropriate response to environmental triggers important for survival and adaptation. There are three major classes of small RNA, with microRNA, siRNA, and piRNA. In the microRNA pathway, Drosha process primary microRNA to precursor microRNA, which are then further processed by dicer to form mature microRNA, which are then loaded onto AGO, and together they execute gene regulatory function. Dicer is also required to process double-stranded RNA to siRNA, and siRNA are then loaded onto different AGO. In some eukaryotes, including C. elegans that I'm studying, this initial recognition trigger an amplifications of so-called secondary siRNA by RNA-dependent RNA polymerase, RDRP, enhancing the RNA response. Finally, piRNA are transcribed from the genome and are then loaded onto AGO or PIWI, and in many organisms, piRNA signals is amplified by the so-called ping-pong cycle, and in C. elegans, piRNA pathway is instead converged into siRNA pathway and involve RDRP. I'll focus on siRNA pathway today. The processing of siRNA require dicer, which form a complex with double-stranded RNA binding protein. In C. elegans, it's called RD4. The two double-stranded RNA binding domain of RD4 is highlighted here, and it plays two major functions. First, biogenesis of siRNA, and second, loading RNA onto appropriate downstream AGO. These functions are very similar to Drosophila R2D2 and LOPS PD. And today I'll tell you how RD4 regulate germline development and C. elegans. But before that, let me just briefly introduce you to C. elegans germline biology. There are two sexes in C. elegans, self-fertilizing hermaphrodites and male. Hermaphrodites contain two gonadal arms. Each arm contain mitotic stem cells, which then enter meiosis. As the developing oocyte passes through the spermatica, it is fertilized, and the developing embryos spend part of their life in the mom and is then expelled through the vulva during mid-embryogenesis. Male, on the other hand, contain only one arm. It starts with mitotic stem cells, which then proceed to form mature sperm. And the male tail is the mating organ. Okay, with that in mind, we found that RD4 hermaphrodite are sterile at high temperature. Instead of fertilized embryo, which are oval and shiny, they lay unfertilized oocyte, which are these dark round cells. And this graph is showing the percent of unfertilized oocyte laid by the animal. We could rescue this defect by crossing the worms with healthy males, indicating that RD4 has a sperm defects at high temperature. We then looked at the expressions of MSP, which is major sperm protein required for sperm function and motility in nematode. In Wombo type, MSP is localized to membranous organelle, is really well organized, but this is really severely disrupted in RD4 mutants. In wild-type males, the DNA in sperm is very well condensed, but in RD4 mutant, we note that some sperm stay loose and some show DNA bridges, indicating chromosomal segregation defects. By RNA-seq, we showed that siRNA targeting spermatogenic genes are upregulated in RD4 mutant at high temperature, which may underlie the sperm defects that we observed. Surprisingly, we could partially rescue the fertility defects in RD4 mutant by expressing RD4 under the control of neuron-specific promoter. So the worms start laying healthy fertilized embryo, and MSP staining also looks much better. These results indicate neuron to germline communication in regulating germline thermal tolerance. So this prompts us to ask how neuronal signaling, especially those involved in environmental perception, affect germline development. We knocked down CMK1, which is a calmodulin-dependent protein kinase homolog important for neuronal gene expression, as well as CGMP gata channel that allows calcium influx into sensory neuron. Both of these mutations partially rescue RD4 fertility defects. In DAPI staining, we can see a lot of accumulations of these large unfertilized oocyte in RD4 mutant and less so in RD4 CMK1 and RD4 TAX4 double mutant. So the animal sensory signaling negatively affect germline thermal tolerance, and we wanted to see if we can pinpoint the exact sensory modality next. And thanks to many, many years of amazing work, starting from John White in the 80s, who assembled the first ever connectome, we know a huge deal about C. elegans neurobiology, including the specifications and differentiations of 60 sensory neurons. Armed with these knowledge, and without going into too much details, I knock out the sensory modality one by one: heat, taste, smell, oxygen sensing, and specific neuron. None of these rescue RD4 sterility at high temperature. But removing a heme containing guanine cyclase, GCY35, involved in oxygen sensing significantly rescue RD4 phenotypes. And here I'm showing you that GCY35 is indeed expressed in a subset of oxygen sensing neuron, suggesting that the lack of oxygen sensing somehow promote germline thermal tolerance in these mutants. We then place the worm in an oxygen chamber at high temperature and examining the germline morphology. In RD4, we normally observe unfertilized oocyte in, it's pretty small here, but I hope you can see unfertilized oocyte stacked in the gonadal arm, and this is not seen in wild type. Consistent with our finding that oxygen sensing negatively affect germline development, we found that growing RD4 mutant at low oxygen level in 7% or 1% mildly rescue the germline defect. So there's less stacked oocyte accumulated in the germline. Interestingly, while it's true that RD4 fertility can be largely rescued by crossing the mutant to males, the brood size is not quite at the wild type levels, suggesting that there might be additional germline defects that are not sperm origin. So we started looking at looking at DNA damage in the oogenic germline using Rab51, which is a repair protein as a proxy. In wild type, this Rab51 signal is most prominent after mitotic to meiotic transition during the onset of recombination, as you can see here. And this is rarely seen in the mitotic stem cell zone in the beginning. In RD4 mutant at high temperature, we see much more of the breaks even in the stem cell mitotic zone. And this, again, is rescued by neuronal RD4 expression. Here I'm quantifying the number of Rab51 foci in the mitotic stem cells zone. Again, you see an increase in RD4 mutant and the reductions in the neuronal rescue strains. Amazingly, removing CGMP-gated channel texture and oxygen sensing GCY35 that I talked about earlier also rescue Rab51 accumulations in RD4, indicating that oxygen sensing circuit regulate thermal tolerance in both the spermatogenic and the oogenic germline, and suggesting that neuronal signaling can affect genome stability in the germline cell non-autonomously. So I hope I've convinced you that neuronal perceptions of the environment can affect germline development during stress. It really had puzzled me for a long time that why and how oxygen sensing circuits could regulate germline development. I think it might be that C. elegans feed on bacteria, which tend to create an oxygen sink, which could be a signal for favorable niche for reproductions, and so the animals pour more resources into germline maintenance when they perceive low oxygen in the environment. With that, I'd like to thank people who had helped me in this project, including Leon, who was a master student in the Rahavi lab, and Sheer, a previous undergraduate students who worked with me. I also like to thank my collaborator, Mario and Hannah in ISTA, and Jonathan and another Hannah in Hebrew University. My postdoc was partly supported by EMBO long-term fellowship. Cool, thank you.
Thank you, Ethan. It was wonderful talk, and it was really beautiful images and stainings, and I was so happy to see it.
Yeah, that was all
Okay.
Hannah, mostly Hannah. Yeah.
Okay, great. Maybe since I will give some time for the participants to write their questions in the Q&A box, I will start with some of them. So, how do you think the neurons are actually communicating with the germline? Like, is there a receptor or this siRNAs are really moving from one organ to another? Like, how it is communicating? What is your hypothesis?
The short answer is we don't know. The
Mm-hmm.
long answer will be as far as so there's a double-stranded RNA channel that known to regulate systemic RNAi in C. elegans called SIT1. And when I remove SIT1, I don't see a change. I don't see at least like the cancellations of the rescue. And so we don't think, at least there's no direct transfer of double-stranded RNA that depends on SIT1. That doesn't say that there are no other receptors that receive these RNA signals if it's RNA. So I know that's not SID1 dependent. Um, I had some results to suggest that neuropeptide might be involved, but I don't think neuropeptide goes from the neuron to the germline, but I think neuropeptide is regulating this oxygen sensing circuit itself. So yeah, so we don't know what actually is being received in the neuron in the germline. So.
Okay. Thank you so much. Yeah. So we have one question in the Q&A box. Is there a relationship between oxygen and temperature, or is it that oxygen sensing pathways are sensitive to high temperature?
Yeah, so I think the temperature aspect of my essay is to uncover the phenotype of the infertility. So I don't think there's direct relationship between temperature and oxygen, but we use temperature as a readout almost. But I can tell you that we recently found these RD4 mutant do have different oxygen sensing responses. We're testing whether they are actually different in different temperatures. So in C. elegans, the most comfortable temperature is 20 degrees, but at least in 25, we saw that the RNAi mutant behave differently. They have different oxygen sensing response. So um, that says that RNAi might be regulating some of this behavior um, at high temperature. So
So the second question is, okay, do wild type animals have similar phenotypes when living in hypoxic environments?
so a wild type, as far as we can see, looking at the germline morphology, they are fine at the oxygen at the hypoxic environment that we tested, which is 1% or 7%. I know the one could get sicker. If you can get lower than 1% for C. elegans. I think you can go as low as like 0.1% or even lower. They get a little sicker, but I don't know how the germline looks like there. But in our hand with 1%, 7%, they're fine.
Okay.
Yeah.
So there is another question from Zanab Afzal. She says, or he, I am not sure. This was really interesting, and I was curious about whether oxygen sensing pathways are different in the different neurons. Just wondering if there are different oxygen thresholds that are sensed by different organs of C. elegans.
Yeah, they are multiple neurons. So I kind of skipped that a little, went through that bit quickly. That there's GCY33 that I tested that guanocycle is also involved in oxygen sensing, but in the different neurons called back neuron. And GCY35 is expressed in a different set of neurons. They all involved in oxygen sensing. Yes, but when it comes to threshold, we didn't test. I don't know enough about the neurobiology to answer that question, how different neuron responds to different levels. But yeah, but yeah, GCY35 not involved, GCY33 not involved, GCY35 is involved, both oxygen related. Yeah.
Okay. So another question I didn't quite understand. Why did you choose to look at neuronal signaling specifically? Are there other pathways in other organs?
Yeah. So I started with looking at the neuronal rescue of RD4, and that gave me a really interesting phenotype. And we start thinking about, like we know that RNAi are involved in sensing the environment. So, for example, worm defective RNAi, they cannot sense pheromone, and therefore they cannot go into a dour state, which is an alternate developmental state. When they get crowded, they'll go into dour, but in RNAi mutant, they can't sense it, so they can't go into dour. So we know RNAi do play play a role in sensory signaling. So putting two and two together, that we have new expressions of RD4 in a neuron, and so we started looking at like environmental perception because a powerful role of RNAi is to collect whatever that's from the outside and interpret it into something, um, physiologically useful for the worms, I guess. So yeah, that's the rationale.
Okay, thank you so much, Ethan. And if there are
Thank you.
no further questions, so a big applause to Ethan for a wonderful work and talk.